use of indwelling medical devices highlights the clinical impact of these infections. The main problem of the use associated with the use of indwelling medical devices is infection, which is usually associated with biofilm formation. Now, what is meant biofilm? Definition, biofilm, we will discuss definition, formation, and regulation. What is meant by biofilm? Biofilms are communities of sessile microbial cells embedded in hydrated matrix of polysaccharides, proteins, and nucleic acid. There are two major polysaccharides produced by Staphylococcus epidermidis: capsular polysaccharide and polysaccharide intracellular adhesion, which is considered as a key virulence factor in the biofilm formation associated with Staphylococcus epidermidis. This figure shows the stages of biofilm formation. We can see that the microorganism at the start will be planktonic cells, which is mean-free planktonic cells. Then it will come, and when it found the implanted device, the first step is attachment, monolayer, then proliferation, microcolony, and maturation. Then after maturation of biofilm and the formation of mature biofilm, there will be phage induction cell death, humoral sensing, which is considered as on-off system, and again the microorganism will be free. That's why the infection caused by indwelling medical devices is relapsing, and that's why, because there is a key on-off system, which is planktonic at first, the formation of mature biofilm. After that, the mature biofilm, there will be disruption and again become planktonic, and another infection will start again. Regulation of formation of polysaccharide intracellular adhesion and capsular polysaccharide, which at first we see that they are the main capsular polysaccharide associated with Staphylococcus epidermidis, is composed of N-acetyl glucosamine in beta-1,6 glucosidic linkage containing deacylated amyl group. The main factor which make regulation for this is ECA locus, which contains beta ADPC gene, which is responsible for regulation of biofilm formation. The role of biofilms and resistant to antibiotics. High resistant to antimicrobial agent is the most important feature of biofilm infection. Infections caused by bacterial biofilms are persistent and relapsing, so very difficult to eradicate. What is the mechanism of antimicrobial resistance of biofilm? يعني ببساطة للبايوفيلم اللي هي associated with the use of indwelling medical devices, usually associated with resistance to antibiotics. First, oxygen limitation and the slow growth, increased mutation and genetic exchange, antibiotic inactivating enzyme and the biofilm matrix barrier, tolerance and overexpression of efflux pumps, induction of biofilm specific phenotype, formation of resistors and the phenotypic variant, general stress response activation, high cell density and the current sensing. Aim of the work. In this study, we will focus on isolation and identification of clinical isolates of Staphylococcus epidermidis from different implanted medical devices. The step number 2 is to investigate in vitro ability of Staphylococcus epidermidis to form a biofilm. Number 3, investigate the impact of biofilm on antibiotic susceptibility of Staphylococcus epidermidis. Material and methods. The first step is isolation and identification. A total of 320 samples from different types of implanted devices, such as fracture fixation devices, central venous pressure catheters, drains, endotracheal tube, and urinary catheters were collected from patients admitted to Sadeghi University Hospitals during the period from May 2011 to November in the same year. All specimens were collected aseptically and transported to the microbiological laboratory at Faculty of Pharmacy, where they were immediately processed and subjected to biochemical tests to be identified. The step number 2 was screening for biofilm formation, and this was done by 2 methods: qualitatively by Falcon tube method and the Congo red agar, and quantitatively by spectrophotometer. Number 3, susceptibility patterns. Sensitivity by disk diffusion. Clinical isolates of Staphylococcus epidermidis were tested for their susceptibility to 11 different antimicrobial agents from different classes of antibiotics. or antimicrobial agent. Step number 2, determination of the MICs and the MBCs for planktonic and biofilm forming bacteria. Results. About 109 isolates of Staphylococcus epidermidis were obtained. And the following figure shows the number of isolated from the different type of indwelling medical devices. About 93 isolates from different medical devices distributed as follow: from 5 isolates from fracture fixation devices from central venous pressure, about 15 isolates, 18 isolates from drain, and 16 isolates from endotracheal intubation with catheters, about 39 isolates. Also, 16 isolates were collected from blood after bacterial infection due to the use of indwelling medical device. In identification, about 100 isolates of staph epidermidis were obtained and subjected to complete biochemical identification as follows. All isolates show gram-positive cocci arranged in bunches or clusters. Catalase test is positive, coagulase test is negative, and the growth at mannitol salt agar was non-mannitol fermented, and it gave gamma hemolytic or non-gamma hemolytic on blood agar. Screening for biofilm formation, it was done by 3 methods: qualitatively by Falcon tube methods and the conjugate agars, and quantitatively by spectrophotometer. This figure shows the colors of isolated colony on conjugate agar media. الفيجر مش واضح قوي بس هو واضح عندي هنا. الcolonies of non-biofilm reducer it give pink colonies or red colonies, while the strong biofilm reducer give black colonies, and from weak to moderate it will give red colony with black periphery. In the second method, quantitatively, it shows the different types, different degrees of color of crystal violet, which show different degrees of biofilm formation. We can see at this non-biofilm reducer, and this was moderate biofilm reducer, and this was high biofilm reducer. The figure shows the number of isolates and the degree of its biofilm formation. We can see that number of isolates and the degree of biofilm formation. About 31 isolates was non-biofilm reducer, 30 isolates were weak biofilm, 90 isolates were moderate, and the 29 isolates were strong biofilm reducer. The third step was susceptibility patterns. Number 8, sensitivity by disk diffusion methods. About 109 clinical isolates of Staphylococcus epidermidis were tested for their susceptibility to 11 different antimicrobial agents, and the results are shown as follows. We can see from this figure the high resistance of Staphylococcus epidermidis to most of the antibiotics used except vancomycin, which show about 100% in the sensitivity, and the next are followed by chloromethnicol, which show 95%, and clindamycin, which show 68%. Then the next step was to determination of the MICs and the MBCs for planktonic and biofilm forming bacteria. The MICs and the MBCs of isolates in planktonic form was done, and the data revealed that none of the isolates under investigation in planktonic form were resistant to the 3 antimicrobial agents. The results were interpreted according to the CLSI 212. This table shows that the isolate number, the MICs and the MBCs values of isolates in planktonic form, which is free for all ratio or all the results are the sensitive. The MICs about 0.5, 0.5, 1, 0.5, and the MBCs was higher than MICs, but we can see that all the isolates in the planktonic or free form was susceptible to the 3 tested antimicrobial agent. While by determination of the MICs and the MBCs for biofilm forming or for isolates in biofilm form, we can see that the results in the table show high degrees, about 512